It looks like it's about time to start, so I think we can get going now. Good evening. I'm Barb Gorgon, co-president of the Minnesota chapter of ARCS Foundation, and welcome to the ARCS Minnesota 12th Annual Scholar Award event. I'd like to welcome all of you from Minnesota, as well as ARCS members from around the country, including our ARCS National President, Karen Ogle from Oregon. So tonight's our chance to celebrate 6 outstanding students nominated by their departments from the University of Minnesota. We especially want to thank 3M Gives for their support of 3 of our scholars from 2021. And tonight we also have the privilege of welcoming Minnesota chapter member, Professor Brenda Ogle, who's our keynote speaker this evening. Please remember to keep your audio muted and screens off during this presentation, and you will have an opportunity for asking questions for Dr. Ogle through your Zoom chat feature in the Zoom bar at the bottom of your screen. So before we start, I'm going to like to give you a quick introduction to ARCS Foundation. It's a national organization and it's had national impact. And ARCS stands for Achievement Rewards for College Scientists, and our mission is advancing science in America through the support of top student research scientists in STEM fields. ARCS started as a response to Sputnik and the space race challenge of the 1960s, but its mission is as important today as ever. As such, it's one of the largest member-based funders of student scientists in the US today. Over $110 million has been given to over 10,000 students in 60 plus years. There are 15 chapters around the country who sponsor talented, motivated American students in 50 top US research universities. Also, ARCS Foundation chapters build industry relationships that support research that's important in their own communities. And 100% of contributions to the ARCS Scholar Award Fund, like here in Minnesota, go directly to students at the University of Minnesota. The Minnesota chapter is one of the newest ARCS chapters, and we've been in existence for over 10 years. Established in October of 2009, and over the past 10 years, we've given out $255,000 to 28 graduate students at the University of Minnesota. The scholars receive $5,000 per year for 2 years, and their funding is unrestricted. Tonight's featured speaker will be introduced to you by Judy Benham, who's co-president of ARCS Minnesota with me and also vice president of University Relations. And she will now introduce tonight's speaker.
Welcome to the ARCS Scholar Award event to everyone who's here. It's my distinct pleasure to introduce our keynote speaker tonight, Dr. Brenda Ogle. She's the professor and head of the Department of Biomedical Engineering, which is one of the departments in which we support scholars. She's also professor in the Department of Pediatrics, director of the Stem Cell Institute at the University of Minnesota, and a strong supporter of the advancement of science in many different ways. Thank you. Barb, back to you.
Brenda, you can share your screen now.
Okay. Great. Well, thank you very much for the introduction, Judy, and thanks to both Barb and Judy for the invitation. It's a pleasure to be with you all this evening. And a big thank you to ARCS for supporting our students for so many years throughout the University of Minnesota, and especially in my home department, the Department of Biomedical Engineering. So tonight I thought what I'd like to share is hopefully something new, something you've never seen before. And I decided to start with a term that I coined recently called responsive medicine. And you might be thinking, oh no, not another precision medicine, personalized medicine, all of this. But the reason that I coined it is because both in my department and in the Stem Cell Institute, what I'm seeing is a movement toward this exciting group of therapies and change in the way that we treat certain disease states. And what this means is a therapeutic or a therapy that, when imposed, responds to the response of a patient to that therapy automatically. And so once you put it in or are dosed with this, it can actually sense the environment of the individual and respond without additional intervention from a clinician. So I'm not going to expand on that much further yet. I just hope that as I'm talking, you see some of the therapies that can actually do this. Most of what I'll talk about is stem cell-based therapies, but there are also lots of categories of medical devices, for example, that are including closed loop interfaces to be able to do something similar. So here's my brief outline so you know what's coming. First, I want to just talk about what are stem cells, then a little bit about how we are interfacing with stem cells at the Stem Cell Institute, some current therapies on the horizon across the globe, and then honing in on some things that are specific to the University of Minnesota and even in my laboratory. And then I'll leave you with some parting thoughts, some things to think about. All the way through, just feel free, as Barb said, to pop your questions into the chat, and we'll have time, I hope, for some discussion at the end. So a really quick primer on stem cells. To be a stem cell, a cell must be able to do two things. First, it has to be able to give rise to indefinitely more cells, has to be able to self-propagate. And it also needs to be able to give rise to specialized cell types, basically the cells in your body that do the work, like a neuron or a muscle cell or a blood cell. And there are many different categories or types of stem cells. I don't have time tonight to talk about all of them. Mostly the types are categorized based on where they're isolated or where they reside in the body, or how we develop them in a dish, or the number of different mature cell types that they can become. So I've decided to talk about pluripotent stem cells because that's what I work with, and that's really what's trending up in the Stem Cell Institute right now. And pluripotent just means that this type of stem cell can give rise to nearly every cell type in the body. And induced pluripotent stem cells are a special type of stem cell. And usually at this point, I ask for a show of hands from the audience to see how many people have heard of induced pluripotent stem cells. And so if you have, please just bear with me for a few minutes. If you haven't, I hope this is an exciting new technology that maybe you've not heard of before, and maybe you can share with those you know to spread the word about this particularly interesting type of cell and its potential for changing the face of medicine. So here's how they're generated. So you can take any cell type from the body. It can be from a skin biopsy, so a skin cell, can be a blood cell. We're finding that nearly every mature cell in the body, when you take it out of the body and propagate it in a dish, you can genetically reprogram it to become pluripotent. And this genetic programming is essentially the overexpression of 4 different genes that allow it to become pluripotent. Once you have those induced pluripotent stem cells, and we call them iPS cells, so now you've got the jargon of the field, they can be expanded in a dish. Remember, that was one of the characteristics of the stem cell, and they can also become nearly every cell type in the body. And the really great advantage of this particular stem cell type is that you can generate autologous cells, meaning induced pluripotent stem cells from living individuals. And therefore, if we develop mature cell types from that stem cell, they can potentially be used as a therapeutic that would not require immune suppression. The other big advantage is that there's no need for embryos. Embryonic stem cells are the other pluripotent stem cell type that have been studied in a dish. So I thought I'd just show you, excuse me, what it looks like when you're changing or converting a cell that was once, in this case, a skin cell into a pluripotent stem cell on the other side. So these are microscopic images. Each of these little flat little things is a skin cell. You give it the new genes of interest. And then I hope that you can see that as you progress along, these cells really change their shape and how they associate with one another. They become much smaller and they start to grow in colonies. You can see at the very end. Then, depending on the environment that you expose those induced pluripotent stem cells to, they can become cartilage, bone, neurons, skin, muscle, as I said, nearly every cell type in the body. And so you can imagine that is a pretty potent tool that could be used for potentially therapeutics that replace damaged or diseased tissue. And so at the Stem Cell Institute, that's really our whole institute was developed 20 years ago. We're actually the very first institute in the country dedicated to stem cell research, you can imagine. So it's pretty exciting that the University of Minnesota had the forethought and the desire to invest in this space. And the whole idea is to study stem cell biology, to better understand the signals and cues that stem cells can respond to become mature cell types so that we can develop regenerative medicine therapies to treat devastating disorders. And along with the research, we're also committed to education and training for stem cell scientists of tomorrow. So that's our building. It's located behind TCF Bank Stadium. And to facilitate our research, the Institute provides core facilities for generating stem cells and for developing new protocols for driving specification of those stem cells. We can do that manually. We also have 2 robots that can automate, in an automated fashion, generate pluripotent stem cells and then differentiated cell types. And we also importantly have a stem cell business incubator. I think this is a really critical time to help companies who are trying to move into this space, to help them de-risk it and also provide a community of scientific support and intellectual power. So we actually have 4 companies that are housed in our Stem Cell Institute. 2 of them are based on technology that actually came from the scientists of our Stem Cell Institute. So it's pretty exciting to see those findings move into commercial products and now clinical therapeutics. We also have training. So in case you know someone who's interested in an advanced degree, we have a master's degree in stem cell biology, a PhD minor in stem cell biology, and also a training program that helps provide support to PhD students. And all of this information is easily found on our website. So I want to talk about the kinds of therapies that are emerging from the Stem Cell Institute. But before I do, I just thought I'd provide a resource for you if you're interested in stem cell therapeutics and what's currently available. This is a wonderful website that's well maintained. The Alliance for Regenerative Medicine shares with you products and the types of regulatory pathways and approvals that each of them have undergone, so that you can be well informed. There are many stem cell therapies that are available that haven't been well vetted, have not gone through full approval processes, and you want to have caution and awareness when it comes to those as well. So I thought I would share, I did a search for clinical trials going on that utilize induced pluripotent stem cells. You can see that there aren't many. This is really, we're in a leading edge of this field. But you can see there are trials going on right now for age-related macular degeneration. So in this case, retinal pigment epithelial cells have been derived from iPS cells to be used for transplant. Also treatment for GVHD, in this case, mesenchymal stem cells derived from induced pluripotent stem cells, dopaminergic neurons to treat Parkinson's, natural killer cells for solid tumor immunotherapy. And importantly, this is a University of Minnesota-based technology. So that's very exciting that we're among one of the top 5 or first 5 therapies. And then heart failure, cardiomyocytes from iPS cells are being used to patch a diseased and damaged heart. So here at the University of Minnesota, I'll just share with you a few iPS-derived future therapeutics. Moving into clinical trials, we have Rita Perlingero, who has derived skeletal muscle precursor cells that can be used to treat muscular dystrophy. We've got Deb Farrington, who is generating retinal pigment epithelial cells. These are in preclinical trials right now, and her goal isn't to use them for transplant, like you saw in that list I just showed you. Her goal is to use them in a dish to determine what therapeutic will be best for a given individual. So she's screening different combinations of drugs to find the best combination for a particular person, because it's been found that there's a wide variety of responsiveness to the therapeutics that are available for macular degeneration. And lastly, we have Ann Parr, who is developing or driving specification of neurons, and her goal is to treat spinal cord injury. So now just a few minutes about my research program. I'm very interested in how stem cell derived cells can be used to treat cardiovascular disease. You probably are very well aware that cardiovascular disease is the leading cause of death in the world. And the biggest problem, and the reason that's the case is this. So you know that the heart is responsible for pumping blood to the rest of your body, and it does so as a function of this beautiful cell type that you see on the right. This is a cardiomyocyte. This is a single cell. You can kind of see the nucleus here in green. These striations are sarcomeres, you might remember from a physiology course, and they are responsible for the electromechanical function or contraction of the heart that allows it to have pump function or capabilities. That's the beauty of the cardiomyocyte. The problem is that when you lose cardiomyocytes due to disease or death, typically caused by ischemia, you don't recover more new cardiomyocytes. Instead, what happens is a different cell type comes in, and this is the cardiac fibroblast. And it does a really good job of very quickly sealing that space so that you don't bleed out. But in the process, it creates basically a scar that is not contractile. So what happens over time is that this kind of scar tissue dilates because it's not able to actively contract, and that's what leads to heart failure in many cases. So it would be great if somehow we could generate new cardiomyocytes, and that's where iPS cells come in. So some of the enabling technologies right now is that we can actually very efficiently differentiate iPS cells into cardiomyocytes in a dish. And so if you want to check out this video, these are some cardiomyocytes that are beating spontaneously in a dish, and they were once actually skin cells from an individual that were converted into iPS cells and then driven to be cardiomyocytes. So that's a very, very powerful technology. And then we learned how to move them from dish to dish. So now you can imagine, okay, now we can potentially inject them into the heart to recover function. And what scientists are trying to understand right now is how to organize them and place them so that they can efficiently couple to existing healthy cardiomyocytes in an individual and then help with pump function. And actually my lab has a couple of technologies in this space as well. But what I wanted to talk to you about was a different idea, and that was an effort to build a living cardiac muscle pump in a dish. And the reason we wanted to do this is twofold. One, we wanted to develop something where we could look at volume pressure dynamics and its impact on healthy and diseased cardiac muscle cells, especially human cells, because actually human cardiomyocytes are quite different from most other animal cardiomyocytes. So it's not easy to use an animal model. Also, we want to move away from utilizing too many animals in our research. And so Molly Kupfer and Wei Han Lin here are graduate students, or were graduate students in my lab who developed this technology. And basically what they did was to develop a bio ink. And that bio ink basically contains only proteins and cells. And they figured out a way to 3D print those proteins and cells, so there's nothing synthetic here, into the shape or form of a mini chambered structure, essentially. And what you can see is that it has an inlet and an outlet, which can be connected to tubing and used to perfuse this structure. And so what we do is we print it. We then allow the stem cells to grow and expand. And then we differentiate them into cardiac muscle so that they can actually pump. And when connected to a fluid source, can actually move volume. The idea is, as I said, to be able to use this as a model in a dish and hopefully one day move toward a bridge to transplant or something living and human that can move blood in the body. And so let me show you a little bit about how far we've come with this model. So up in the upper right is just showing the 3D printed structure. And I should tell you that what we did to establish the digital template. So, you know, most of you, I'm sure, are familiar with 3D printing. So 3D bioprinting is the same thing. It's just that you're using biological components like cells and proteins. So you have a digital template, and in this case, we used an MRI scan of the heart. We scaled it down to the size of a mouse heart. And then we printed it with our bioink layer by layer until we had created the whole structure. We modified it slightly to be able to create a one-way flow loop. So we basically deviated the septum between the ventricles to allow blood or a media to flow through. So this is what it looks like. Let's see if I can get this going. This is one of our structures, and we call them HCHAMPs, which stands for Human Chambered Muscle Pump. You can see, in this case, liquid flowing through the structure. Here, I'm showing the thickness of the wall. This is a microscopic image of the wall of this chambered muscle pump, and this red that you see indicates cardiomyocytes. And if we zoom in a little more closely, you can see the nice striated pattern of these cells. So again, these were all derived from human-induced pluripotent stem cells. And they can function together to actually contract spontaneously. We can also electrically stimulate them to contract at a frequency that we impose, and you can see that here. This outline is an HCHAMP, and this what you'll see is contrast associated with the movement of voltage. This is a voltage sensitive dye. So you can see it progressing along the structure with each beat. So at this spontaneous activity is starting from this side and moving to this side of the HCHAMP. Then we decided to pace it at the other side, and you can see much more robust activity coming from the other side. So with that, I'm going to wrap up because I want to make sure there's time for questions. But my parting thoughts are basically, I hope that I've shared with you some new things tonight, and a look into certain types of responsive medicine that in this case utilize stem cells and what we're understanding about the biology of stem cells in a dish. We're learning how they might function after transplant, and that's exciting. There's a really great new institute at the University of Minnesota that we partner with called the Institute for Cell, Gene, and Immunotherapy, abbreviation CGI, which is helping us to further move stem cell therapies into the clinic, helping with regulatory hurdles especially. Speaking of regulatory therapies that are not approved by the FDA or other reputable regulatory agencies should be avoided. We're still definitely at the early stages of advances, but one of the things that we consider along with sort of the excitement of new technology is how we're going to develop these new therapies through the lens of health equity, and considering carefully how we study these cells and tissues in the context of all populations in our world, and think about the outcomes and how they benefit uniformly our world. So we benefit from a knowledgeable community of scientists and non-scientists alike, and we're always at the Stem Cell Institute looking for engagement from the community on this and all aspects of what we do. Because of that, we're thinking of concrete ways to incorporate diversity, equity, and inclusivity lenses in biomedicine. And we've been fortunate enough to be able to hire a new DEI coordinator, Gabby Horsford, who is helping us do just that, trying to help us build a diverse biomedical community and also to carefully evaluate the ways that we approach biomedicine. And so with that, I want to thank you again, thank ARCS, and I'm happy to take any questions that you may have.
Well, thank you, Dr. Ogle. We do have one specific question about your work, and that is, tell us more about the genetic reprogramming via, how do you do that to grow those specific cells from stem cells? You mentioned you expose them to specific environments, but could you give us a little more insight on that?
Sure. So there are two parts. I'm not sure if you're interested in the reprogramming to become an iPS cell or the differentiation piece to mature cell types, but I can say a little bit about each. So for step one, we call it reprogramming because you're taking a mature cell like a skin cell and converting it to an iPS cell. In that case, we have things called genetic vectors, and essentially they are pieces of DNA that encode those particular transcription factors that we know need to be turned on to convert a cell to a pluripotent stem cell. So we literally pipette a solution that contains those genetic vectors and get them into the cell, and then the cell will use its own machinery to convert those gene transcripts into protein. So that's how the reprogramming happens. And then to get them to become mature cell types, that's a really big, hard, challenging question. It's all about the environment that the cells are exposed to that drives specification. And we get those clues about what those cues could be from studying development. So all of the years of developmental biology and everything we've learned, that's now being used to drive specification of stem cells in a dish. So for us, we're learning that extracellular matrix proteins, and you know these by collagen is one, elastin is another one. These proteins actually provide more than structure. They're actually potent signaling molecules that can help to drive specification. There are other signaling molecules that can do the same thing and are sort of redundant in their function. So for every cell type, it's taking years for us to uncover the cues that will drive specification. But most major tissue types are moving along pretty rapidly in this space.
Okay, thank you. So another question that's in particular about cardiomyocytes. You had stated that human cardiomyocytes are different than animal cardiomyocytes. Can you just expound, tell us a little bit about that?
Yeah, so there are some big things like heart rate or beating frequency. Small animals like rodents, what we typically work on, are beating at frequencies of like 4 to 6 hertz, whereas we beat at about 1 hertz. And part of that is enabled by the differences in the structures of their sarcomeres. Without going too far into the weeds, there's differences in isoforms like myosin heavy chain 6 versus 7 that allow for some of that difference in functionality or ways that they actually need to function. So it's differences in isoforms to accommodate the function of that particular organism.
So just two more questions. The first one is pretty high level. And how many grad students are in your department? And do you focus, primarily on this work on cardiomyocytes, or how do you decide what to work on next with the grad students? Because you have just this, plethora of opportunities.
Right. So in our department, we have about 150 graduate students who work in at least, we have 22 primary faculty and then another more than 50 affiliated faculty. So the graduate students have a large, broad range of biomedical engineering projects that they can work on. And they get to select this by selecting 3 rotations in 3 different labs when they first start in our program to be able to decide what they want to work on. And we pride ourselves in really prioritizing the interests of the graduate student as much as possible when they're matching to their research lab. In my own lab, I have 10 graduate students, and I kind of do the same thing. I write my new ideas coupled with their new ideas to obtain research funding. And then those ideas are what sort of drives a lot of what we do. But it's also informed by and guided by the interests of the graduate students themselves. So that's how we decide. And of course, we're motivated by the end goal, which is how do we get closer to translating this into something beneficial for society.
Okay, thank you. Finally, we know that you have your interests in improving the heart. But what do you think, you see this whole field, what do you think are the most exciting emerging areas for breakthrough knowledge for regenerative medicine in the next few years? What other things are you really excited about that could come about either at the U, but maybe in other places?
Right. I think the biggest one is the one that I talked about, which is the any cell therapy that utilizes iPS cells as a source, just because of the ability to avoid immune responsiveness. But there are many other things that we learn from stem cells, like so that we don't necessarily need to transplant the cell. We're really understanding in a dish some of the factors that are secreted to drive a certain function, and those are really leading to great advances in new drugs and drug delivery. And then finally, I'd say the other really exciting thing is all of the new gene editing tools that are coming online, really making the idea of gene therapy, especially for monogenic disorders, that means one gene mutation or change causes a disease state. So I think gene therapy kind of took a spin in the late 90s and had some setbacks. And I think a lot of the new technologies that are being developed can address those and have been very valuable in the research front, and I think are going to move into the clinic pretty rapidly.
Thank you so much. That was obviously very, very interesting talk. And we at ARCS Minnesota are really privileged to be working with you and some of your grad students. So thank you so much. And now we'll go on to another exciting part of the program, which is to actually introduce our ARCS scholars.
Well, thank you so much, Ingrid and Dr. Ogle, especially for learning about this fascinating area of research. It's just an exciting time, and I'm sure the discoveries are coming quickly and very exciting to share this information with us. Just to let everybody know, we are recording this session, and we'll send the link out afterwards. So if you want to review some of what Dr. Ogle said, you'd have that opportunity. So as Ingrid said, now we're going to move on to our Scholar Award section of the program. This is the time where we honor our scholars. I want to review a little bit about what who these scholars are and how they're picked. They're selected by their departments. They look for academically outstanding, high potential graduate students in STEM fields who have innovative research projects, and many of our students also have additional contributions outside of the laboratory. Before I introduce our scholars, I thought I would just run through some of the impact that the Scholar Awards have on the students. In a national survey from 2020 of ARCS Scholar alums, 91% said their Scholar Award had significant importance to them as students. And 94% said it was important for their moral support and encouragement. 93% said important for prestigious recognition. 85% important for focusing more time on their research. And 81% important for their completion of their degree. And then likewise, once ARC scholars are in their careers, this is the impact that they have had now on science. They are mentoring future scientists. They're making discoveries and innovations. They're communicating the importance of national scientific competitiveness, and are founding or leading organizations that have had significant scientific or economic impact. So these are very important contributions. Now it's time to introduce our Scholar Awards for 2021. We have we have these scholars span 6 different departments at the University of Minnesota: neuroscience, molecular biology and biophysics, biomedical engineering, civil, environmental and geoengineering, computer science and engineering, and mechanical engineering. So we have 2 second-year awards going to Courtney and Joe, and first-year awards are going to Judy, Mariah, Mark, and Matthew. And each of these students will receive a check for $5,000 that they can use as they need for their studies or general support. So first, I'd like to introduce Courtney Hutton Pospick, and she is receiving a 3M Give Scholar Award. She's in computer science and engineering. Courtney?
Hey everyone, I'm Courtney, and I've lived all over the country. Right now I am in Minneapolis. So in my undergrad, I studied mathematics and writing at Wellesley and Occidental, and towards the end of my degree, I had the chance to conduct research over the summer at a local lab that dealt with virtual and augmented reality. And I fell in love with the variety in the field. We worked with medicine, education, robotics, and collaborated with a lot of other disciplines. So after graduating, I started my PhD in computer science at the University of Southern California. And I worked with the same advisor and lab that I had researched with as an undergraduate student. And after the second year of my PhD, I followed my advisor here to Minnesota to continue my degree at the U. So my current research also deals with virtual reality and augmented reality. And specifically, I look at new ways that people can interact with the space around them. And I work on developing adaptive technologies that help people of all types with all sorts of physical capabilities interact with the technologies that we're able to use. So initially, I only conducted research in virtual reality, but while running a user study, I was puked on by 2 participants. And this led me to switch to augmented reality, where users are much less likely to get motion sick. So one of the things that I love about my work is showing people new impossible spaces and ways to interact with the world. And as was mentioned, this is my second year as an ARCS scholar, and this award has been a bigger blessing than I ever imagined. I originally hoped to form new relationships with the community of scholars and to buy new equipment for my research. And the first part of that came true, and I've really enjoyed the discussions I've got to have in the last year with my peers in the program. But what I didn't realize was that 2020 had another surprise in store for us and my family. and my husband was laid off shortly after I received the Scholar Award. So this award helped us pay our bills and stay afloat while he found a new job. So I really want to thank all of the donors for making this award possible. It's made more of a difference than I ever would have expected. Thank you.
Thank you, Courtney. Okay, our next scholar is Joe Emerson, who is the 3M Gives Scholar Award. And again, this is his second year, and he is in the neuroscience department. Joe, you can go ahead.
Yeah, hi, I'm Joe. I'm a third year PhD graduate student in the graduate program for neuroscience here at the University of Minnesota. I'm originally from Shelby, Ohio, and I earned my bachelor's degree in physics and computational neuroscience from Ohio Wesleyan University in 2019. Before studying here at the U, I participated in research that focused on understanding the mechanisms of epilepsy. And this experience really solidified my interest in using mathematical models to better understand the brain. I'm currently working in Dr. Cheryl Ullman's lab on research in the human visual system. And my research project focuses on linking data from functional brain imaging technologies, including functional magnetic resonance imaging, or fMRI, with computational models to better understand early visual processing in the primary visual cortex. My project is focused on understanding how visual areas in the brain are able to interpret our visual environment by leveraging context or spatial context. This has applications to the study of psychotic disorders and is aimed partly at understanding the neural mechanisms that underpin visual hallucinations and distortions in schizophrenia. Articulating early visual processing and computational models will allow us to make specific predictions about the causes of visual abnormalities and will hopefully contribute to a better understanding of underlying causes of symptoms of schizophrenia. The prospect of understanding the mechanisms of our perceived experiences and then using this knowledge for applications to treating psychosis continues to motivate me in my research. And this ARCS Scholar Award has really helped me achieve my scientific communication goals. And last year, by facilitating my attendance at several conferences, hopefully also going forward as we transition back to more in-person conferences. And in addition, it's opened up just a broad network of scientists working on a diverse set of fields. And I really want to thank the donors for making this opportunity possible.
Thanks, Joe. Our next scholar is Judy Sharon. She's a Minnesota Chapter Scholar Award winner, and she is a first-year scholar. Judy?
Hi, everyone. I'm Judy Sharon. I'm a fourth-year PhD student in the Biochemistry, Molecular Biology, and Biophysics program. I'm originally from the Bay Area in California, where I did my undergrad at Berkeley, where I studied microbiology. And after my undergraduate degree, I went to the USDA and a few different startups to do research on soil microbiology, where we were working on solutions to move away from synthetic fertilizers. When I finally did come to the U a few years ago, I started to work with Dr. Kate Adamala to build cell-free biological computers. All this really means is that I'm trying to tie together Boolean logic from electrical engineering and computer science into really simple biological concepts like DNA transcription and RNA translation into proteins. The hope is that this kind of foundational work in the near or distant future will be able to help us build hopefully really highly integrated prosthetics, or help us program bacteria better, or even help us build biological supercomputers. And right now, the Scholar Award, especially since I'm in the latter half of my PhD and trying to figure out what I want to do after after graduate school, the Scholar Award is going to help me attend conferences and workshops that will facilitate career development. And because of that, I want to sincerely thank all of the ARCS members and donors that are making this all possible. Thanks.
Thank you, Judy. My cursor disappears. There we go. The next scholar is Mariah Dorner. also a Minnesota chapter scholar, receiving the first year of her scholar award. She's in the Civil, Environmental, and Geoengineering department. Mariah?
Hi, everyone. I'm Mariah. So I grew up in Hugo, Minnesota, and from there I attended the University of Wisconsin-Eau Claire for my bachelor's degree, where I studied biochemistry and molecular biology. So my undergraduate research involved some studies on the degradation of azo-dye pollutants, as well as some work in computational enzyme kinetics. Following my graduation from the University of Wisconsin-Eau Claire in 2014, I spent about 5 years working before I came back to graduate school here at the U of M. So during this time, I contracted as a fermentation research technician at Cargill. I worked as an asbestos and fungal microscopist for Pace Analytical. And finally, I found a position as a research technician in the Department of Plant Pathology here at the U of M. And from there, I transitioned to the graduate program in civil, environmental, and geoengineering. So my fascination with the intricacies of biochemical processes and complexities of microbial community interactions, combined with just an overall passion for the development of technology to better protect our environment and human public health, have all inspired me to pursue the work that I'm doing today. So I'm now a third-year PhD student in in environmental engineering. I work in the Barron's lab investigating the effects of biochar on nutrient removal processes in activated sludge systems. So biochar is a carbonaceous solid similar to activated carbon. It has sorptive and electron shuttling properties that have been shown to enhance microbial transformation processes. So my work involves the operation of bench scale sequencing batch reactors that are amended with biochar. And these reactors are meant to simulate treatment processes that occur in full-scale wastewater treatment plants. So with these reactors, I perform chemical analyses and microbial community characterization to elucidate how biochar affects their function. And because previous research has also shown that biochar can enhance the degradation of toxic chlorinated pollutants, I plan to expand my work to elucidate the effects of biochar on the degradation and fate of contaminants of emerging concern that enter our wastewater treatment systems. So with that, I would like to thank the ARCS Foundation and its donors for their support. It's a huge honor to be recognized among so many amazing young scientists. And I really look forward to building a relationship with the ARCS Foundation, its donors, and fellow scholars for years to come. And I'm excited to see what I can do with this award and where it takes me. Thank you.
Thank you to Mariah. That's wonderful. And our next scholar then is Mark Gotthelf. And he is a 3M Gives Scholar Award winner for the first year in the Mechanical Engineering Department. Mark?
Hello, my name is Mark Gotthelf, and I am a fourth-year PhD student in the Mechanical Engineering Department here at the University of Minnesota. My research is on understanding the effect gender bias has on ratings of a surgeon's technical skill. This work is part of the larger narrative of promoting diversity, equity, and inclusion, which is being addressed across fields and industries. Bias in the evaluation of what a student's or person's skills can result in minority groups being at a disadvantage in their education, regardless of their actual skill level. When I first came to the University of Minnesota, my original intention was to get a PhD in rehabilitation engineering, but during my second year, I started talking with my advisors about how certain department practices were affecting my mental health and potentially biased towards certain groups of students. My advisor let me know that he was submitting a grant to address bias in surgical skill evaluation and that I could do my PhD on the topic of bias, something I didn't realize was possible as a mechanical engineer. I immediately accepted and loved doing this work. My interest in addressing bias comes from my undergraduate experience in promoting diversity at a primarily white institution, working to teach middle school girls about computer science to promote gender parity in STEM, and being a queer, non-binary engineer myself. In addition to my research, I have been the co-lead of our department's diversity committee, and in January will switch to being the co-lead of the College Diversity Alliance to further promote these efforts. Outside of engineering, I enjoy spending time with my family that consists of my wife, 3 cats, and 2 birds, watching terrible movies with my friends, playing music, and enjoying nature. Oh, and I am also a huge nerd that at 27 still loves playing the Pokemon card game with my friends around the Twin Cities. I would like to thank the ARCS Foundation and all the donors for this generous award and the opportunities it will allow. It is an honor to be recognized by my department and ARCS.
Thank you, Mark. Appreciate that. And then the last award goes to Matthew Rennis, Richard Pyle, MD, Memorial Scholar, donated by Judy Benham in honor of her late husband. He is in the Biomedical Engineering Department. Matt?
Hello, everyone. My name is Matt Renesh. I am a graduate student in the Biomedical Engineering Department, working in the Biosensing and Biorobotics Lab Group, led by Professor Suhasa Korandarmaya. My background is in neuroscience, which I studied here at the University of Minnesota. And my graduate work now is focused on uncovering changes in brain-wide activity during spatial learning in mice. Previous research has elucidated a system across many regions that forms a system for spatial navigation and decision-making in animals, but questions remain about how brain-wide states arise out of this system. And another question that arises are how these regions are coordinated to produce one behavior. Much of the research that has been done on this system has been done while imaging from individual regions using a variety of techniques and technologies, but it hasn't really been performed across the brain at a high spatial and temporal resolution in an unrestrained animal. And this is mainly due to the lack of available technology capable of performing such imaging. So to perform this in our lab group, we develop a miniaturized fluorescence microscope capable of brain-wide calcium imaging in freely behaving animals. And this is one of the core things that our lab group does. We develop technology for neuroscience as one of the things at the core of one of my beliefs is that I believe technology drives innovation. And this is, I think, fundamental to some of my research motivation. And I think there is precedent in history to this view as well, where influential scientists and engineers over the ages who were at the forefront of major advances, where they were typically the first people to master or even create emerging techniques and technologies. And this dates back to people, well, a couple of examples I love to use are people like Galileo, who heard of technology for telescopes from some 2 other engineers, and he built a better one of his own. And then he used that to study the stars, or more pertinent to my field of study, a physician in Germany by the name of Corbinian Brodmann, who used his microscope to identify, just looking at the cortex of the brain of postmortem patients from the hospital, and just looking at how the differences in how they were organized, the cells were organized, he was able to find over 50 brain regions. And the list can continue. So I believe advancing technology and adopting it will lead us closer to discovering all of the mysteries that lie within the brain and other biological systems, and that these discoveries, as well as the technology itself, have the potential to greatly improve life for many people in the world through clinical applications, social, societal, and infrastructure advances, or through research, which is where I stand on this. This has always motivated me growing up, and the people who hold the beliefs similar to it as well, figures such as Carl Sagan, these types of people who advocate for this influence me, and my brothers who always had a desire to describe things in nature to the best of our ability, but we fell short on the big questions. Where does consciousness arise? Where does the universe come from? And part of my sort of itch to understand this, to figure this out, was scratched where in my undergraduate degree, where I studied neuroscience, I became aware of research using some of these tools, using some advanced imaging tools, recording tools, to figure out and discover, make large breakthroughs in our understanding of the brain, such as population control of movements, which was led by some of the people who are with us in Minnesota. And so I don't want to close my introduction without stating how grateful I am to be and honored to be selected as an ARCS Scholar 2021 awardee. I think the award will greatly impact my ability to continue my research, present it to the scientific community, and it will also help me work to disseminate the technologies that I've taken part in developing. Also, the community that I'm being invited to, I think will be a great resource for reflecting my ideas and discussing with the greater scientific community. So I look forward to the beginning of a very fruitful relationship. Thank you.
Thank you, Matt. Appreciate you doing that. And congratulations to all our scholars. We're very proud of you, and we're looking forward to getting to know all of you as we go through this year. In summary, I'd like to say that it's ARCS Foundation's belief that support of STEM education is essential to U.S. economic growth and technological competitiveness, and it helps to ensure continued U.S. leadership in global innovation, health, and quality of life. ARCS scholars have gone on to positions of leadership and distinction in academia, industry, and government efforts. 9 out of 10 ARCS scholars work in their sponsored fields, sharing knowledge, developing technologies, launching startup companies, inspiring youth to pursue the challenge and study and career in STEM fields. These students will provide the answer to some of our country's biggest problems and represent the future. So 100% of the scholar donations coming to Minnesota go to the Minnesota Scholar Award Fund, and your support is needed now more than ever. So how is it that you can help ARCS? Well, there are several ways. First of all, you can get more information about ARCS Foundation Minnesota chapter, and we'll be following up with this with links to our website and opportunities for you to get more information. Women may consider joining as a member of our ARCS chapter. Again, we will provide you information about how to do that if you're interested. And men can be friends of our ARCS Minnesota chapter through donations to the ARCS Foundation Minnesota chapter. And you can attend our ARCS Foundation Minnesota events, such as this one. Also help spread the word about ARCS Foundation in Minnesota to your family, friends, colleagues. We have an opportunity coming up that will be quite exciting. It's a don't miss event on November 18th. We're honoring Dr. Michael Osterholm as ARCS Minnesota Scientist of the Year for 2021. This will be a unique opportunity to see and hear Dr. Osterholm. We will both have in-person and online opportunities to hear this event. He is a nationally recognized expert on the pandemic. He is the director of the Minnesota Center for Infectious Disease Research and Policy, known as CIDRAP. He's a Regents Professor, McKnight Presidential Endowed Chair of Public Health. He's been interviewed by numerous national news organizations for his expertise on the pandemic. So you will be following up with information about this event in email, so please be sure to keep an eye out for it. The in-person event is capped at attendance, and so make sure you get your reservations in early. So before you go, we'll provide a link to the video recording of the webinar, and we'll send that out in a couple days. If you want more information, you can go to our website, minnesota.arcsfoundation.org. And I appreciate your participation tonight, and thank you, and look for more events from us and opportunities to hear from our scholars.